Cancer is an internal conflict. Natural selection within the body favors rapidly growing cells, which develop into tumors and overwhelm the interests of the individual. But exactly what kind of conflict is cancer? To answer that question, it's useful to do a historical overview of the ideas of conflict. In the 1970s, Egbert Lee argued that the Mendelian 50-50 segregation ratio in meiosis is actually the outcome of repression of competition between chromosomes within the genome. Because we don't see meiotic drive, in spite of the fact that natural selection continuously favors competition between chromosomes and meiotic drive, the absence of observed meiotic drive in most cases means that there must be mechanisms in the genome that repress competition between the chromosomes. And so the normal Mendelian segregation ratio of 50-50 is really the outcome of repression of competition between chromosomes that's ubiquitous within genomes. Around the same time in the 1970s, Richard Alexander was interested in the evolution of human sociality. And he noticed that over the history of human social evolution, that at first, early in evolution, humans lived in small groups of closely related kin. And so kin selection and close genetic relatedness was a sufficient explanation for group cohesion and cooperation within human groups. But over time, human groups grew to be much bigger. And as human groups grew to be bigger, Alexander realized that that meant that the degree of genetic relatedness between individuals within groups necessarily got reduced. And so the question then became, what maintained internal cohesion in societies as the degree of genetic relatedness in kin selection got reduced within groups? And Alexander argued that moral systems and laws actually served to repress internal competition within groups and promote group cohesion, social cohesion within groups. And that social cohesion within groups helped groups to compete against other groups in group against group competition. So we see by the 1970s that there were well-developed ideas of the critical role of repression of competition within groups in order to form cohesion among the members of the group and make the group function more or less as an integrated unit. But although those ideas were well developed in the 1970s, for the most part, very few people paid attention to them, and they were really confined to just a few experts. The next big event in the history of ideas was in 1987 when Leo Buss wrote a book called The Evolution of Individuality. And Buss was very interested in the origin of the germ-soma separation in multicellular organisms, because Buss noted that there were many independent evolutionary events in the history of life where there was a separation in development between the germline cells that would be transmitted to the next generation and the other cells in the body that are not transmitted and are called the somatic cells. And this germ-soma separation evolved repeatedly in the formation of multicellular individuals. And so Buss was curious about that phenomenon. And Buss's argument for the importance of the germ-soma separation was that if the germline is separated early in development from the other cell lineages, and the cells that will be transmitted to the next generation for reproduction are then separated out and privileged in early development, then there's no way for competition between cell lineages over reproduction to arise. The other cell lineages that are not in the germline cannot reproduce and be transmitted because they're physically separated out from the germline. And that physical germ-soma separation represses the potential for competition between cell lineages. And so the somatic cells that will not be reproductive are essentially a sterile caste. And the only way for them, those cells, to increase their own fitness is to aid the fitness of their genetic relatives, the genetically related cells in the germline, which is by raising the fitness of the individual as a whole. So the repression of competition between cell lineages then integrates the interests of the different cell lineages and prevents competition between cell lineages. And Buss argued that that was a fundamental aspect of the transition to complex multicellularity and the formation of individuals, hence the title of his book, The Evolution of Individuality. The next year, Maynard Smith, after reading Buss's book, said, Buss has identified one of the great problems in evolutionary biology. How do higher level units evolve that repress the competition between individual members within the group, such that the higher level acts as a new unit in evolutionary biology, essentially a new individual? And Maynard Smith said that's a great problem in evolution, and one we don't fully understand. And so this is something that really needs greater attention. So Maynard Smith agreed with Buss about the importance of the problem, but Maynard Smith disagreed with Buss's explanation for multicellularity. Maynard Smith said the way in which life has solved this problem of cooperation among cell lineages within multicellular aggregations is the single-cell zygote. If all of the cells in an individual arise from a single common cell, the zygote, then all of those cells descend from a recent common ancestor and are genetically identical or very closely related. And that very high level of genetic relatedness between the cells in an individual means that kin selection is a sufficient explanation for the cooperation among cell lineages and the lack of competition between cell lineages. And so we don't need to invoke this more complex explanation of repression of competition. And so Maynard Smith basically understood that if mutations arose within individuals, that could potentially break down the degree of genetic relatedness within the individual and lead to competition. And that was Buss's argument. But Maynard Smith said that if cells are descended from this recent common ancestor, mutation is too weak to create enough mutation pressure within an individual lifetime to favor a lot of competition between cell lineages. So Maynard Smith agreed with Buss's overall argument about the problem, but disagreed with him about the particular explanation. And so we see the tension between kin selection and repression of competition as alternative explanations, Maynard Smith coming down on the side of kin selection for multicellularity. And we can see eventually that when we get back to cancer, we have this same problem, because cancer is competition between cell lineages within a body, and how are we going to understand that? But for now, we're just focusing on the history of ideas to set the broader context. And we have a pretty good context, but we're missing one key idea about internal conflicts within aggregations in order to understand cancer. And what's that idea? Well, in order to get to this next key point, let's talk about a particular example that motivates the idea well. And so we're going to think about microbes. Microbes often start with a small number of cells and grow into a large clone. So that's very much like multicellularity, starting with a single cell and growing into a large population of cells that are closely related. So let's focus attention on a particular scenario. Imagine that there's a bunch of sugar and a single bacterial cell lands in that sugar and starts to use those resources, and so it divides, the cells divide and make a large population. And bacterial clones can grow into very large populations. So we have then a high degree of genetic relatedness because all of the cells descend from a recent common ancestor, the single cell that founded that clone. We can call that the zygotic ancestral cell in this case. And if we actually follow such a process in real bacteria, what we often see is that as the clone grows up and gets larger, inevitably mutants arise that can grow faster than the original progenitor cell. And those mutant cells then form overgrowths that overgrow the original ancestral cell. And so that's just like a cancer. The cancer is basically mutations in cells, cause those cells to divide faster, and those cells overgrow the individual with a tumor that expands out. So the inevitable overgrowth that happens in bacterial clonal growth is essentially conceptually identical to a cancer. And typically in bacteria, when we get those overgrowth mutations, cells that divide faster and overgrow the original ancestral clone, they're also less efficient at using resources. So they grow faster, but they have a reduction in efficiency. And we can measure efficiency as how many grams of sugar does it take to make a gram of biomass, of bacterial biomass. And the faster growing clones are less efficient at converting sugar and resources into biomass. And that lower efficiency reduces the fitness of the group as a whole, because now we've got cells that are dividing quickly, but they're less efficient at using resources, and so their final population size is actually going to be lower when they use up all the resources. And so we see a trade-off between the rate at which the cells grow and their efficiency. And the key trade-off in terms of conflicts is better expressed in the following way. It's a timescale conflict. The short timescale of rapid growth within the clone overwhelms the longer timescale of efficiency of the clone in producing total numbers of bacterial cells that can compete against other clones. So the short timescale of growth within the colony overwhelms the long timescale of competition between colonies. This timescale conflict is one of the great features that shapes the architecture of life. And the key historical reference here is in 1966. George Williams wrote a book called Adaptation and Natural Selection in 1966. And the fundamental point of that very famous book is that timescale conflicts dominate life. That there's always a conflict between reproduction on the shorter time scale and success on the longer time scale. And that almost always the short time scale overwhelms the long time scale because the short time scale happens more often and the intensity is greater. Selection among cells within a clone happens continuously and immediately. Competition between clones is something that happens less frequently and more slowly. So the shorter time scale overwhelms the longer time scale unless there's some special explanation at the higher level that represses competition or through kin selection. Williams recognized that sometimes there is some special explanation at a higher level, but his argument is that in the history of life that's very rare, and that almost always we should assume the short time scale dominates unless we have a very clear understanding of what special attributes are happening on the longer time scale. And we can see that Maynard Smith and Buss and those other arguments previously all fit within this context. The argument between Buss and Maynard Smith was, Maynard Smith said that kin selection is sufficient because the time scale over which mutations arise is not long enough within an individual body. And so there will not be mutant overgrowths within individual bodies for most multicellular aggregations. Buss said that's not true. There is enough mutation pressure, and therefore you need repression of competition through germ-soma separation. But we see timescale conflict as the fundamental organizing principle. And I think that remains true today as the best way to think about these problems. So let's think a bit more about this bacterial clone story because it's interesting. In terms of the bacterial clonal story, the pressure for overgrowth is powerful because bacterial clones grow to very large population sizes, and there is enough time and enough cells such that mutant overgrowths are expected and are a ubiquitous, powerful force. And that means that inevitably and frequently and overwhelmingly, there's a selective pressure that pushes for rapid overgrowth and overwhelms the interests, the higher level interests of the original progenitor cells. That means there's constant selective pressure on bacterial genomes to repress the origin of mutants that can overgrow the clone. Now, how can bacterial cells repress the origin of mutants? Well, there's a number of potential mechanisms within the genome of bacteria that can reduce the origin of novel mutants that have the the properties of being able to grow faster, but perhaps less efficiently. And so this is something that really hasn't been looked into very carefully, and I think this is one of the great unstudied problems, because I strongly predict that bacterial genomes do have mechanisms that tend to repress the origin of rapidly growing mutants, and that this could be studied and would lead to a very nice research project that would eventually lead to, I think, an excellent research publication. So this is an open problem that we come across when we think about cancer more broadly in terms of internal conflicts. Let's follow that line of thinking a bit more about how we can take these concepts and think more broadly about natural history beyond just the narrow scope of how we usually think about cancers in our own bodies. We'll get to that later. But for now, let's think about one more example. Another example is insects that carry symbionts. Many insects carry microbes, bacteria, sometimes fungal microbes, that the insects carry within their bodies and transmit through their eggs from mother to daughter. So an insect might have a lot of bacteria in its body, and some of those bacteria in a female will get into the eggs and be transmitted to the next generation. So the symbionts are transmitted vertically along the matriline. And traditionally, it's thought that because the symbionts are transmitted vertically along with the genes of the host insect, that there's no conflict of interest between the bacteria and the host, that vertical transmission is sufficient to reduce conflict of interest. But that's not true, and we can see it already from the things we've talked about. For example, the population of bacteria within the individual host, if there's a mutant that arises among those bacteria that cause that mutant bacteria to outcompete its neighboring bacteria and get into the germline and transmit at a higher rate, that mutant will spread. It's like an overgrowth mutant or like a cancer. And so then that reduces the success of the host as a whole. So again, we have that same tension that we see in cancer as an internal conflict. Now the host can do a few things to reduce the probability of getting mutant overgrowths among their symbionts. They can reduce the number of symbionts that make it into the egg, like a zygotic bottleneck, single-cell bottleneck, and that increases the genetic relatedness among the symbionts within the host by resetting how recently the cells have descended from a common ancestor. And with high genetic relatedness, there's less pressure for mutants to overgrow their neighbors. So one prediction is that hosts will impose genetic bottlenecks on their symbionts. Another aspect is that the host can impose a germ-soma separation. The host can set aside a small subset of the symbionts for transmission through the eggs early in development and prevent the other bacteria from accessing the eggs and being transmitted. And so the other bacteria within the host are essentially somatic cells that cannot compete for transmission because they're physically separated out from the germline. And so we see this same tension between kin selection and germ-soma separation arising in the history of insect symbionts. And there's a lot of opportunity here to study how insects and symbionts actually evolve under these pressures. It's an opportunity to study these same conflicts that Maynard Smith and Buss were arguing about, but in a different context. And so a little bit has been done on this, but there's still great opportunity for comparative studies and further publications that haven't been carried out yet. So now we have the basic concepts of internal conflict, and we can see that the key driving factor in cancer and in the germ-soma separation is the rate at which mutants arise that can potentially overgrow their neighboring cell lineages, the degree to which mutation pressure is sufficient to overcome the single-celled zygote in close genetic relatedness, and therefore requiring another level of repression of competition because kin selection is not sufficient. Excuse me. And so the question is, how powerful is mutation pressure in an aggregation of growing cells? And so we can just think about our own bodies as a good example. You start life with a single-celled zygote, and the cells in your body descend from that single cell, and so they're all closely related. But how much mutation actually happens in building your body? Well, that depends on how many cells are in your body. And how many cells are there? There's about 37 trillion cells in your body. And of course, in producing your body, some cells die. So let's just say about 100 trillion cells have to be produced, and actually probably more to make your body. 100 trillion is 10 to the 14th. Now, how much mutation happens? Well, let's focus on a single protein coding locus, a single gene. And the typical story is that each gene mutates at about a rate of 10 to the minus 6 per cell division. So now the question is, how many cell divisions are there to make the 10 to the 14 cells in your body? And that's an easy calculation, because every time a cell divides, you go from 1 cell to 2 cells. So every cell division adds 1 cell. So to add 10 to the 14th cells requires 10 to the 14th cell divisions. So if we multiply 10 to the 14th cell divisions by a mutation rate of 10 to the minus 6 for the mutation rate for a single gene, then that means that every gene in your genome mutates about 10 to the 8th times in making your body. And that's just for a single gene, that's not over the whole genome. That means there are 10 to the 8th or 100 million mutational events at that single genetic locus to make your body. Now, there are even more cells that carry those mutations, because if a mutation happens early in development and it's neutral, then all the descendant cells will carry that mutation. And so a large number of descendant cells carry that mutational event. So there are going to be many more than 10 to the 8th mutated cells at that single genetic locus in your body. And then, of course, you have many gene loci on the order of 10 to the 4th, and many other parts of the genome that are non-coding that will mutate. So the mutation pressure and the amount of genetic variability that's generated in producing your body is immense. In fact, the evolutionary history in your body to produce those 100 trillion cells is greater than all the vertebrates that have ever lived in the history of life. And so the evolutionary history in your body is huge. So going back to Buss and Maynard Smith, I think it's quite clear that the potential for mutation pressure is immense within the body. Of course, it's not really what matters is not mutation, but what matters is the origin of genetic novelty that can create new traits that can cause overgrowth. And so finally, that brings us to the question of cancer. And so now let's actually look at cancer within our bodies, within this broad context. And so the issue is, you start with 1 cell, and your body's going to grow up to 37 trillion cells, a lot of cell division, and you get a cancerous overgrowth if a novel trait arises that causes cell lineages to grow faster. We'll return to that issue of what actually happens to make cell lineages grow faster. But for now, let's just review the main conceptual framework in modern cancer genetics by which we understand cancer. The main idea is that as your cells divide, mutations arise in some cells that cause those cells to divide faster. Those are called oncogenic mutations. Mutations happen in oncogenes. Oncogenic mutations cause cells to divide faster. There are other types of genes that repress the formation of tumors. When your cells are not acting normally, they typically will die. They invoke a program called apoptosis, and they kill themselves, which is very effective at protecting you. Also, there are some genes that function as checkpoints on the cell cycle and keep the cell cycle from just rapidly going and reduce the potential for rapid cell division. And in cancer, we know that there are mutations in these oncogenes which accelerate cell division, and mutations in the tumor suppressors that knock out the protective effects. If you have cells that are dividing faster and are not dying normally, then those cell lineages expand and form tumors. And that is the essence of the modern theory of how cancers get started. And essentially, almost everybody who works in cancer subscribes to that genetic theory for the origin of tumors. But there are some problems with that theory that are quite clear and suggests that there must be more to this story. A tumor is more than just a bunch of rapidly dividing cells. A tumor actually has to create a variety of novel traits. Tumors are actually the origin of novel tissues with many new functions. Tumors have to evade the immune system. Tumors have to attract the blood supply. Tumors have to secrete proteases that break down the tissue barriers so they can spread through the tissue. And tumors are often created by a variety of different cell types working together. So they're not just mutations in a single cell. So the origin of a tumor is actually the origin of a complex new tissue with multiple functions. And so we have to think about more than just mutations. We have to think about, where does novelty come from? How do new traits arise? And this puzzle of the origin of new traits is perhaps the greatest puzzle in all of biology. If we think about Darwin, we think about what was Darwin really doing and thinking about in his time and in his books. Of course, his most famous book is The Origin of Species. But if you read The Origin of Species, the book's not really about the origin of species. It's about the origin of novelty. The question in Darwin's time is, where do new traits come from? How did birds come to fly? Where do our eyes come from? Where are these new traits coming from? What is the creative force? So it's the origin of novelty that Darwin was concerned with. And in this cancer problem, it's the origin of novelty that's really the key problem that we need to think about in the origin of tumors. Simply invoking genetic mutation isn't sufficient. Now, the typical view in modern evolutionary biology is that new traits arise. When a mutation arises, it's advantageous, it spreads. Another mutation arises, it's advantageous, it spreads. But the origin of a complex tissue really raises some other questions. And so let's look at some recent studies in cancer biology to understand the origin of novelty and where new traits come from. And we'll set the broader context here. So I want to tell you about some studies that have been done in the laboratory on mice over the last few years in lung cancer, because this really opens up, I think, some new views on how cancer arises and new views on the origin of novelty in general. So in a laboratory, a particular group of investigators, they engineered mice to carry a KRAS mutation. And KRAS mutations are oncogenic mutations that are thought to make cells divide faster. And KRAS is known to be one of the common mutations in the early formation of lung tumors. So they engineered these mice to have KRAS mutations, and they grew up the mice to see what would happen. And what happened was, mostly not very much. Yeah, there was a little bit of maybe a little extra cell division, but not really even significant tumors forming, and nothing that was really cancerous or invasive. So then they said, well, okay, in most lung adenocarcinomas, there's also often P53 mutations. P53 mutations are the most commonly mutated gene in human cancers. And P53 are tumor suppressors. They're thought, for example, they do many things, but one of the key roles is that P53 plays a role in cell death when cells are damaged or not behaving correctly. And most tumors have to knock out that cell death program so that the cells don't kill themselves and the tumor can spread even though it's abnormal. And so they made mice that had KRAS mutations and P53 mutations. And they looked at what happened in those mice, and they grew up the mice, and what happened was almost all the mice got lung cancer. So that matches perfectly the genetic theory of the origin of tumors. You get an oncogene, you knock out a tumor suppressor, you've got faster cell birth, you've got less cell death, you get an expanding clone, you get a tumor. So at first glance, that seems like a perfect support for the common theory of the origin of tumors. But what was interesting about this study is that they used single-cell technology to actually look at individual cells and try to understand the causal processes. And when they looked at the cells that just had the KRAS mutation, they didn't see any real extra-cell division, right? No tumors formed. But at the single-cell level, they could see changes in gene expression. They were looking at the epithelial cells that line the lung, and normally those cells express the gene program of lung epithelial cells. But with a KRAS mutation, those cells began to express other genes that they don't normally express. And they found 12 different cell states. A cell state is just a set of genes that are being expressed at any point in time. And they noticed that the cells were kind of wandering over these different cell states stochastically. So some of the cells were expressing maybe some pancreatic genes that are normally expressed in the pancreas, but aren't expressed in the lungs. Now the cells are not pancreatic cells, they're still lung cells, but they just express some of those pancreatic genes. And so there were a variety of different gene programs that were turned on that are not normally turned on in lung cells. And what was interesting was that there was a particular cell type that was kind of in the middle of this fluctuating set of cell types that had the following properties. The cells, the genes that are normally turned on in trophoblast cells were partially turned on. Trophoblasts make placentas in pregnant females. Placentas are the perfect kind of tissue if you were building a tumor. Placentas grow fast, they attract the blood supply, they reject the immune response. So a lot of the traits that are needed to make a novel tissue that grows into a tumor are expressed by trophoblast cells. Now the cells weren't placental cells, they're still lung cells, but they're expressing some of those genes. They express some of the genes of chondroblast cells. Chondroblasts make connective tissue, like cartilage and other things that hold tissues together. If you're going to make a new tissue, you've got to hold it together and give it some structure. So the chondroblast program could potentially be useful. And these cells also express part of the tubular kidney epithelial cell program. Tubular kidney epithelial cells are particularly good at rejecting immunity. And rejecting immunity is one of the most important attributes that a tumor needs to acquire in order not to be killed by our own immune system. And so what happened was that in forming the tumor, that those different gene programs were recombined to make a totally new kind of cell type that was the tumor, that used parts of those different gene programs. And so the origin of novelty actually came from recombining different components of cell plasticity. So we see that initially there was a genetic mutation, it was KRAS, but KRAS didn't trigger rapid cell division. What it really triggered was the release of developmental terminal differentiation, the release of plasticity. And so then when you add in p53, you get even greater release of plasticity and dedifferentiation, because p53 plays a role in normal development of lung cells. In normal lung cell development, there's a cell type called AT2, which is like a cell that can develop into other cell types. It's very plastic. It's used to heal wounds, so it's very flexible in being able to remodel tissues. And that AT2 transits into the AT1 epithelial cells that line your lung when p53 is expressed. So p53 plays a key role in differentiation to make terminal differentiated lung cells. When p53 gets knocked out, there's a transit back towards the AT2 type, which is more flexible in terms of its cellular plasticity. So these key genetic mutations, KRAS and p53, seem to be functioning primarily by releasing the plasticity that creates the novel tissue. So the genetics is important, but as a releaser of developmental plasticity. The developmental plasticity plays the creative role in making the novel phenotypes. And then there are subsequent secondary genetic mutations that stabilize those novel phenotypes. And so the origin of a tumor is genetics releases plasticity, plasticity creates novelty, and genetics stabilizes that novelty. That's a very different view from the traditional view of the origin of novel traits. But it's essential because remember that the driving force in internal conflicts is the origin of novelty, because it's how fast new traits can arise in the short term that can overwhelm long-term interests. And it's not just mutation pressure. Here we see that it's actually the actual generative mechanisms that create novelty, which is a much richer view for understanding the origin of tumors and for understanding internal conflicts and timescale conflicts in general. So that's the story that wraps up and links all the different things that we talked about. There's one more research project that I think is worth mentioning as a final comment here, and that is that there's quite a bit known about the mutation rate of tumors in different stages of tumor development. One of the things that often happens in tumor progression is that the internal mutation rate within the tumor rises early in progression. And that rise in mutation rate seems to be important for releasing variety, for maybe releasing plasticity, maybe creating new genetic mutants that have new traits. And so we see an increase in genetic mutation rate that's common. Sometimes the mutation rate is very high early in the formation of a tumor. I would actually say that it transits past the mutational meltdown threshold. So if the mutation rate is so high that the tissue might break down if that mutation rate stayed high for a long time. But what happens is that there's a temporary burst of mutation and then a secondary stabilization that reduces the mutation rate. And so we get a trajectory of mutation rate and stabilization through the formation of a tumor. And understanding that more deeply is really an open question and a great research project. There's quite a bit known about it already, but not enough known that I think we really understand the full story in the context of how timescale conflicts and the origin of novelty shape internal conflicts in general and tumors in particular. Thank you.